tonight is on Our webinar series tonight is on training for low concentration targets versus residual odour. And we're going to look at some of the current research and training implications. And before we start, I'd like to acknowledge the traditional owners on the various lands that we meet tonight. I'd like to recognise elders past, present and emerging, and acknowledge any First Peoples that are joining us tonight. So our webinar was pre-recorded mainly because our speakers are all based in America, and it was going to be very, very difficult to convince them to get up at 4 o'clock in the morning to talk to you guys. So I pre-recorded this webinar last week, and so you'll be watching the pre-recorded webinar. And if we've got any particular questions that we would like to send to our speakers, they're quite happy if we put them into writing. We can email them, and then I can get back to you if you've got anything particular. So we have 3 speakers today. We've got Paul Bunker, who is the founder and principal of Chiron K9. He's a canine consultant, which is a canine consultancy company specialising in detection. Paul served for over 22 years in the British Army's Royal Army Veterinary Corps as a canine trainer, instructor, and in 2000.
Sonja, you're muted.
Sorry, guys. There's just one thing with Sonja's. Won't be a moment.
Should we start again?
Yes, please.
All right, we'll start again. Okay, so, all right, so starting again. So tonight's webinar series was looking at training for low concentration targets versus residual odour. We're looking at the current research and training implications. We'll be joined by Paul Bunker, Mallory DeChant and Bart Rogers. And the webinar was pre-recorded because all of our speakers are based in America, and it was very difficult to convince somebody to get up at 4 a.m. to meet with us at 7 o'clock at night. So I pre-recorded this webinar last week. So if anyone's got questions for our speakers, they're very happy if we put them into writing and we can discuss them at the end of this webinar. And I'll be getting back to them via email with any questions you've got, and then we can post them either on Facebook, their answers, or onto our website. Before we probably start, I'd like to acknowledge the traditional owners of the various lands we meet tonight. I'd like to recognise elders past, present and emerging, and acknowledge any First Peoples that are joining us tonight. So our first speaker that is joining us is Paul Bunker. He's the founder and principal of Chiron K9, a canine consultancy company specialising in detection. He served for over 22 years in the British Army's Royal Army Veterinary Corps as a canine trainer, instructor and assessor. In 2017, he formed Chiron K9 to concentrate on detection dog research and conservation and environmental detection. He's been credited with thousands of confirmed finds of targets during his career. He is an author and co-author of several published papers and wrote the workbook, Imprint Your Detection Dog in 15 Days. We'll also be joined by Mallory DeChant from Texas Tech University. Mallory was awarded her Bachelor of Science in Animal Science from the University of Findlay in 2017, her MS in Animal Science from Southern Illinois University in 2018, where she studied the physiological impact of simulated search work in search and rescue human remains detection canines. In 2019, Mallory completed her certificate in canine fitness and conditioning from the University of Tennessee. Mallory completed her PhD in animal science at Texas Tech University in 2021, where she studied various canine experience factors that impact detection dog performance. Currently, Dr. DeChant is a postdoctoral research associate at Texas Tech University, where she's studying nutritional solutions for enhanced cognition and olfactory discrimination, reduced core body temperature, and muscle damage or inflammation in working dogs. Our last speaker is Bart Rogers, and originally we had planned to have his wife Lucia join us, but after seeing the questions that we had posed, Lucia decided that Bart was going to be a better fit. So Bart Rogers is the Chief Canine Instructor of Auburn University's Canine Performance Sciences breeding program. Bart oversees the development and training of candidate detection dogs from early development and socialization through advanced odour detection training. Bart also plays an integral role in training and operating dogs for various canine odour detection research projects, predominantly related to explosives detection and wildlife conservation. So here we go. I'll press play and hopefully this all works. People let me know if it's not working. Okay. Started. So welcome everybody. So we have Paul Bunker, Mallory DeChant and Bart Rogers with us today. And I suppose we might kick off. So just asking each of you in turn, I'm not fussed who goes first, but I suppose our first question would be, what research have you done around low concentration odour? So who wants to kick off with that one? Do you want to go, Paul?
Yeah, I'll go. I think a lot of the work I do does involve low thresholds of odour, whether that's on purpose or not. So sometimes even in the training process, I'm going to be dealing with low thresholds, and particularly in the hydrocarbon detection, the oil spill, I have to work on very low thresholds because the dogs find it way too easy if I don't. And in fact, within the training lab, even small amounts will just swamp the lab with odour. And for those of you that have read my workbook, you know, one of the principles I teach, the three-leg stool, is that trace amounts of odour are important to calibrate the dog's nose in detection. Specifically in research, there are two major research projects that we did. One is with Dr. Nathan Hall's team, and it was out of Texas Tech University, but Dr. Barnes's lab, and we were looking at Bellingers, invasive muscle Bellingers, eggs, within water, and can canines screen sample waters from lakes and actually detect Bellingers in comparison to an eDNA? And if they can, what's the equivalency of those two detection methods? That was really interesting. You know, these Bellingers are microscopic small. No way can you see with a human eye. And I was actually surprised how effective the dogs were at detecting Bellingers. And I don't think the actual full paper's been published yet. It certainly is soon. But the dogs were detecting 0.825 Bellinger per milliliters of water. And these are microscopic, obviously. And the other one was out of Dr. DeGrief's lab at Florida International University with Michelle Kopinski. And we were looking at fractions of oil and what the dog detects specifically within a crude oil. So they fractioned it out, collect it onto a medium, and then mailed it down to me, and we investigated those mediums and reported which the dogs investigated or not. And they were in, you know, parts per million and parts per billion captures that the dogs were detecting. So really low thresholds. But beyond that, the dogs have demonstrated that they've detected things outside of technology capability that we had at the time within the research for the technology to even detect. So below those levels, we've no idea what the dogs are detecting.
Yeah, interesting. Very interesting. So many questions already. Mallory, do you want to have a go?
Sure. So I can think of the two studies that we've conducted on low concentration. So the first one did with Paul, actually looking at spontaneous generalization to low concentration. And for that one, we saw about a 10-fold spontaneous generalization from what they'd previously been trained to. And then the other one that we did was we kind of call it the old school lineup now, with all of our fancy equipment that we have. So just regular old PVC pipe on wood, ports like that. And we explicitly train dogs to lower concentration. So we started at 10 to the -2, which is about 0.001. And then we diluted it further, liquid dilution and mineral oil. And then we tested their threshold. And so we found that when you explicitly train dogs to lower concentration, you can get about 100-fold improvement. Looking back at that study, I wish we would have went further to see how far we could really optimize their threshold for that odor, but of course we ran out of time. But yeah, so with explicit training, we did find 100-fold improvement for their threshold.
Yeah, that's good. I've done something similar, which we might chat about at some stage too. Bart, you've done quite a bit.
As going on right now, not completed, from the Naval Research Laboratory, I believe we have a machine that's a TVGen. It's a trace vapor generator. So it's basically the machine that's used to calibrate the sniffing machines at the airports and things of that nature. But then they've engineered it to present odors in a way to our dogs. So in piloting that, if we're talking about low thresholds, we used an odor just to pilot with, and this would be more for explosive work, but this is a non-explosive odor that we used, and we actually got stable detection in the parts per quadrillion with that. Now, as every odor varies, and other odors were tested, one odor in particular, dogs are not able to detect it as well as the odor we previously used, but definitely in the low parts per trillion. The problem that we run into most of the time, I guess you've probably seen this as well, is the dog will start finding, in whatever apparatus you're using, when you start doing low thresholds, they're going to find something to associate to be efficient that will be not the actual target odor you're training on. So when you start seeing, you start dropping threshold and you start seeing the increase in performance, or the dog, oh, this is not as hard as I thought, which we've had basically redesign the machine and control for everything so that we know that it's odor. But the dogs basically, it's very hard to see dogs' threshold and to train to working operationally relevant threshold sometimes without them associating something other than the odor that you want them to respond to. And from an operational perspective, not so much published study, low thresholds, I mean, a lot of times we train in a way that is not going to be successful in operations. For example, like training for pythons. Every time we imprinted on live pythons were stressed. When I found one in an operational environment, they were not stressed, which was a lower threshold of odor. And so we really had to work on that and drop that the dogs would work in an operationally relevant way to be successful.
Yeah, I think we see that in search and rescue dogs as well, where the difference between stressed and unstressed animals. And yes, good. Yeah, very interesting. One of the things that I've been thinking about a little bit is that whole, when we're working with extremely small amounts like parts per trillion, just knowing that the dogs aren't just giving us an indication for the sake of, like you said, being efficient, because it's such small amounts, it's actually hard for us to have equipment that can test the accuracy of that as well. And I think that's really hard in a laboratory situation, much harder we've all got labs, so we've all got equipment that can measure these things, but for people who are training dogs and trying to train them onto low threshold, it's actually really difficult, I think, to know when that odor's there and when it's not there, and when the dog's actually alerting to the odor or just alerting because we think it's there, I think, as well. So we've actually really covered what's the lowest concentration targets you've trained on. Does anyone want to sort of add to that? I suppose we've already sort of said what sort of concentrations we've added to. So I think one of the big ones that I've been thinking about is, can you guys discuss masking and overshadowing? So masking, one of the issues, Bart, you've probably seen this a little bit with the Python work, is that we do a little bit of frog work as well. So Zoos Victoria do frog work. When I say we, I'm the collective we of everybody in Australia that I know that's doing work. They do a bit of frog work, and we've found that reptiles don't have a huge amount of odour, or some reptiles particularly, and then when they're buried underground, the possibility of that masking of ground. I'm looking to do a bit of work also around soil type and wetting, whether that makes a difference. So I was wondering whether you guys could maybe make a comment about when odours are masked. And Paul, you were sort of talking about the water samples, and I know water doesn't necessarily mask, but might actually enhance if a way. But can we have a bit of a chat about that? And then maybe we'll have a chat about overshadowing separately.
I think the first consideration is, and you kind of raised it, is that when you bury something or something buries itself, such as a reptile, it's not the same odor anymore. So it's not necessarily masking, it's the way the odor can move through the environment for the dog then to be able to detect it. And what it does detect, both in quantity and quality, is a different odor than the target that is not buried. The first way that any target odor is going to be affected by the environment is to move through the substructure, and that's going to be initially through any channels that exist, such as in sand or some silts. And that's going to give a pretty close headspace type delivery, although the quantity is going to be lower. But moisture is extremely important because it's the movement of moisture through soil, silt, clay, that transports the molecules of your target through that substructure. Otherwise, there's no way it can get to the surface. There's only two ways. Well, the third way is through pressure, which would be like an oil leak in a pipeline or a body physically forcing the odor through the soil. But if we think of something that's passive within a soil environment, then the moisture is going to be significant. But now you're delivering an odor on the surface, and I call it a footprint, which is the dilutable molecules within a target, and the undilutables are held below the target surface. The only way they can get through to the surface is through physical movement. And that would be either the water actually picks them up and physically moves them through, which takes some force, or we turn the ground or probe or something similar. So when something is buried, we first of all have to understand that the quantity and quality of that target headspace is not the same. And you must train the dog on that headspace, which is from a buried target, and not necessarily have an expectation that just because you train on the target, your dog is going to find something that's buried.
Yeah. Good. Interesting. Bart, do you want to have a go?
So from buried targets, I have very little that I can speak about experience from conservation. My stuff wasn't typically underground, other than finding nests and things of that nature. But I agree with him 100%. Like I hear masking and things like that. I don't know about anything masking or other than it, like Paul was saying, the odor changing as it makes it through the surface and how it makes it through the surface. Whether, I mean, I've had dogs find things that even if they were able to transfer from training without doing buried training work with the target, they may be hitting what was the headspace, as Paul would say, but it may be a root following a root of trees up as a path of least resistance, which could be 8-10 feet away. But I haven't seen anything of so much as impossible. It's just what you trained on is different than the odor that is buried, that is available to the dog. Yeah, I don't have anything other than masking, except for what I explained with, the odor of the python can change. When we dealt with the reptiles like that, if they were in ambush mode, they don't want to have a smell. They want to be able to ambush prey. They also don't want to be found by predators.
Yep.
So the odor was different.
Yep. Mallory?
So I haven't done any buried studies, or really any masking studies as well. We do have one study that is in the works where we're looking at mixtures, but it's not the primary objective of that study. But it is a component that we are taking into effect, of course. But again, I haven't done any, conservation studies or anything buried.
Yeah, we might come on to mixtures in a minute with overshadowing as well. I suppose the other question that goes with the masking is our DAF, which is Department of Forestry, Fisheries and Agriculture. Here, one of the issues that we have, and I suppose this comes into that masking, one of the issues that they have is odours that are coming in sealed in foil containers. And they were finding that the dogs were having difficulty with that. And they thought that that there wasn't odour coming through. But we sort of think, and again, we're going to probably start looking at this, that it's more that they need to train on odours in those containers. What do you think about that? Do you guys, anyone done anything along those lines?
So with containerized stuff, in my experience, is the question is everybody, a lot of times you go threshold. Okay, if it's permeable, everything's somewhat permeable. But like Paul was saying, the water soluble molecules versus not in that context. Well, in some barriers, what part of the odor signature our molecules are able to penetrate through that barrier versus some that are not. So if you present and you work with the raw, pure target, what part of that is key to the dog yielding a response or a strong change of behavior that when is completely concealed or containerized is not readily available or is not available until a period of set time that it can penetrate that barrier. So not versus a threshold or low amount making it out or portions that do not make it out.
Yeah. So that,
Yeah,
I suppose.
I agree with that, absolutely. And again, I go back to the three-legged stool within my workbook. It's why I imprint using discrimination exercise of mixing your target with other materials, because you're not necessarily going to get a nice clean headspace presented to the dog. You're going to get parts of headspace and you don't know which parts, and also different materials will present different parts. So you have to clearly communicate to the dog, this is your odor profile, and it might be presented in portions, it might be presented in different quantities, but the quality will change. And as long as the dog understands that broad spectrum of sometimes this particular molecule is present and sometimes it isn't, but it's still my target. I mean, some of this will come into generalization, obviously, with conservative dogs, liberal dogs, etc. But we must present to the dogs a clear understanding of what presentations of headspace are. And just like Bart was saying, any container can filter, if you like, filter out certain molecules and only present others. So I agree, it's not necessarily a threshold issue. It's what is the odor that the dog is receiving? Does it recognize it as target? Or is it so specific, it thinks that's not my exact target, so I'm going to ignore it?
Mm. Yeah.
I think we can add to that is like Paul was just saying, is we always think about pure and pristine in training. And I haven't found anything yet operationally that was pure and pristine. So, go along with what you're saying is, I like to say is, I'll give you the full library of what is potentially in that odor for a few reps, but I'm going to vary everything all the time so that I could somewhat I've reinforced scenarios that could be the scenario I never think about. This type of container, this type of barrier material mixed with this. So I have my cleans, I have my dirties, I have things I find in the environment that I will containerize things with. And if the dog misses it, he misses it. I can tell you what I'm gonna go back and work on then.
Yeah, that brings us to what Mallory is talking about, which is basically working with cocktails and mixed odors, doesn't it? Mallory, do you want to add what you're doing with your cocktails and mixed odors?
Yeah, I think it's different because we're in a laboratory, right? So it's a very controlled setting, just like Bart said. It's not, outside in the real world operational. Using the olfactometers, it is very clean, and we know what headspace we're getting. We know quantities, everything like that, concentration. So it is different, but and we've only tested a few dogs right now, so it's very, in the beginning stage. But we're just looking at one target odor with varying, mixtures with distracting odors, and how the dog's performance do. Again, it's a secondary aim for it. The primary one is looking at a nutritional solution to improve and optimize the dog. But it is something, the few dogs we've tested, we've seen certain odorants are overshadowing that target odor. But again, when you think about these dogs, they have very limited experiences in training with mixtures. So their primary training is, that single odorant, whatever they're training to. But again, it's very different because it's a very clean environment versus operational in the field.
Yep. So I did a research project where we trained dogs. We took 12 that were already detection dog trained, and 12 raw dogs, and trained them on a cocktail. So 4 odors mixed together, and then tested them on the single odor. And we wanted to see whether a few things, overshadowing. So we saw a little bit of overshadowing with one of those odors. But one of the things we found was that we thought that the ready trained dogs would actually generalize a lot better. And we actually found 0 difference between dogs that had been trained on single odors and then trained on a cocktail. And then when we separated the odors out, they were perfectly fine to find them. And the raw dogs that had never been trained on a single odor, we trained them on a cocktail. When we separated them out, they found those single odors as readily as the trained dogs. So yeah, it's an interesting one, but we had a bit of overshadowing as well. So one of those odors, they were less able to find due to, we think, overshadowing. So so talking about overshadowing, is there any particular substances that you find are difficult when you're working with low concentration odors, like in particular substrates or particular, I suppose, substances that cause overshadowing within training?
I can only, just to follow up with what you said about the cocktail method, it hasn't been published or put out yet, but using Dr. Hall's boxes, the olfactometers, we did a study of generalization, and now given the type of explosives we used and the range that we use from plastic explosives down to a chlorate or nitrate, and their signal strength, we did get overshadowing in the way that our cocktail group, they only learned the loudest odor when we separated them. They did not generalize or know, if I remember correctly, any of the other odors, except for the strongest one when we bundled our cocktail method. Yeah.
We got ours learnt all 4, but they weren't reliable on one of them. So yeah, I think
I'm not
it
saying
depends.
that you wouldn't get it if you classified them differently and how you cocktail or bundled them, but just for the experiment from loud to not loud odor that we know, they only learned the loudest.
Yeah, and I think that would be the case with like conservation odors as well. Like if you're looking at trying to mix reptiles with bats or something like that, or you wonder whether they. One of the things that we were discussing here with one of our other dog handlers is we train our dogs on foxes, because foxes are an invasive pest here. We train them on fox scat because we put out baits or traps. So we find in toileting areas, and they, what I've heard from a few handlers actually is a lot of the foxes tend to eat cat scat. So dogs that are trained on fox scat, that where the foxes have been eating the cat scat, the dogs will find not only fox scat, but they'll actually find straight cat scat as well. Have you guys seen that sort of stuff within your dogs, or?
I've trained scat dogs, but I didn't train on any species that ate other species scat. So I know I haven't experienced that one.
Yeah. But have you ever heard of where you've got something like where an animal's eating a particular diet, and then
Yes.
they're finding the things that they're eating as well. So have you've
Yeah,
seen that?
I had I trained on zoo bear
Yeah.
diet on black bears to go to the southeast for a population study and stress and all that. And that doesn't transfer. They're looking for zoo bears in their diet. So, and then the bears from away from households versus near households, that was a whole different thing, because at the time that we transferred there, they were eating blackberries. So might as well have trained on blackberry jam.
Yeah.
So,
Yep, good, interesting. Yeah, we certainly one of the things that we're looking at here is the fact that, yeah, if diet is really important with their ability to pick up particular odors, and they'll generalize to certain populations because of their diet. So residual odor. So is residual odor just very low concentration to the dog? And therefore, if we want dogs to ignore residual odor, are we in fact teaching them to ignore low concentration? Possibly influencing detection when odor is masked. So this is a question from Naomi. So when she's talking about frogs again. So if we want them to find the frog, not where the frog sat on the rock. And so she wants to know, yes, whether if we're training them to ignore residual, are we training them to ignore low concentration as well?
I think for me, it depends how you're collecting your target. If you're collecting via swab, then you're training residual. Because ultimately, when you swab a reptile, you're collecting the excretions on the skin predominantly rather than the VOCs from the reptile itself. And those excretions are what the reptile is leaving as it moves through the environment. So you're taking residual off the body, presenting it on a medium, which means it's now a residual. That medium, whether it's a gauze or Whatman paper or whatever you're using, Get's End, is no different from some reeds or some moss. It's a medium that holds an excretion odor of the target. So the first thing I would ask is, how are you collecting and presenting the target odor? If you want to differentiate between residual within the environment and the target, then collect the headspace of the target and see if that has enough variance that the dog is able to discriminate and know the difference between the two. And I think significantly with reptiles and frogs, that is an important step that people have to understand. Now, you may get away with swabbing a frog and finding a frog. Whether that's generalization on the dog's part or natural transfer to, that's interesting, I'll go and check it out. I get a reward for the headspace now. And you're pairing during the training process of a generalizing dog, a liberal dog, and you accidentally train the dog through that process, or whether actually the swab does give enough information for the dog to find. But I would definitely be on the side of collecting VOC and discriminate residual, as in excretions, if that's a problem that your dog is showing interest or responding all over the place on excretion.
Yeah.
I'd like to add to that with the project that we did here early on. I liked what he said. You accidentally trained the dog. So the Burmese pythons in the Everglades, we didn't know what odor, and we couldn't get samples here because invasive species, other than pet trade, borrow the snake. So we use swabs. Like you say, and taught the dog residual, taught the dog task, how to work using that, transferred, then got live snakes. So then we actually have odor, headspace, snake in a bag, snake in a box, snake in a field. But the dogs were really keyed to, since we use residual, on the residual odor left in an area, which as we started finding snakes, or even before we started finding snakes there, it was a problem from a selection of type of dog issue, because we taught the dog residual and we would be on canals and areas. They may not have been there that day. They may have been there the day before, given the weather conditions. Well, the dog would encounter residual. We couldn't tell if that was. We didn't know if we had transfer learning, strong change of behavior. And one dog, a couple dogs couldn't handle that constant pressure. One dog could. But the beauty of it was once we got it where they found the live target but could tell residual, I could give information once we proved the dogs were detecting presence up to 2 weeks. Because to get in the park and path, I got given a 2-mile stretch of road to search it, tell them what I thought. And dog said snake went halfway across the road, turned and went back into the mangroves, and they showed me a picture, and I tied my flagging tape to where they had pulled the same branch flagging tape off. So the dogs was real good at finding the odor on the ground. But what that did was for the snakes that we couldn't pull out, because they were detecting snakes underground, they were detecting snakes underwater, respond and stare at water, and we couldn't get to them, then we could use all that information to then organize traps, people to monitor those areas that those dogs were saying of heavy traffic, of a lot of presence. So for every snake we found, we took out numbers more just from the information the dog gave us without us obtaining it in that moment.
Yeah, I think that's important as well, having that, particularly with monitoring as well and trapping and things like that. That gives you information about where to set up. For conservation as well, it will give you information where to set up trail cameras and things like that.
Yep.
We
coordinate a lot of efforts to get information.
Yeah. Mallory, do you want to add anything?
I think that was very well said. I don't really have anything to add on residual. We did a study, if you're talking about low concentrations and wanting to have dogs not alert to a certain concentration, that is possible through explicit training. So we've identified, their spontaneous generalization and said, we no longer want you to respond to this low concentration. So through explicit training, you can do that, but again, very different than what we're talking about in the field.
Yeah. Yeah, there's been a lot of talk about training dogs in a specific concentration range. So not so low that it's residual, not training them on high amounts as well. So particularly in our customs, people have been talking a little bit about that, training within a concentration range. Yeah, and the difficulties of finding that. But again, it's a little bit about the right dog, I think, too.
Well,
So
one thing, I mean, just bouncing this off since we've got everyone here. When I think of residual, I think of residual as odor on a surface.
Yep.
Okay.
Yep.
And not so much a threshold that I present to a dog that then creates that.
Yeah. It's the odor that's left behind, isn't it? Yeah. Where it's touched, which is what Paul was saying, it's whatever you would swab off on your skin that you leave behind when you touch an area. Decomposition of odors, have we like thinking about again, dogs that are finding odors that are possibly looking at tracking and moving through areas where past odors have been, and that decomposition, do we think that affects whether a dog is excited about our odor or not, whether they can tell the difference a bit between old odor and fresh odor? Any comments to make on that?
Yeah, they definitely can. And Mallory did some research with us on training dogs actually to ignore older odor and only respond on fresh odor. So it was crude oil. We wanted the dogs to respond only on fresh spilled oil and ignore natural oil, which a natural aged oil, which exists. We're on the Gulf Coast where we are, all over the place. I mean, we went out a week last Monday and the dog had 33 finds of weathered oil buried and on the surface in a small survey area that we use. And I've actually had over 1,000 in the same area. And then we train 2 specific dogs, which are with citizen scientists. They go out and we've taught the dogs to ignore that natural oil and only find the same type of crude oil, but a fresh example. So we know we can obviously train dogs, and we do the same with scat. When you don't want aged scat, you only want fresh scat. So the understanding of a dog in the difference between an old target to ignore and the fresh version of that target, that's perfectly feasible. And just like I always say, you know, for me, the one sentence that sums up training is every behavior has a consequence, Thorndike's law of effect. And if the consequence of ignoring an old odor is as rewardable as the consequence of detecting a fresh odor, then you're going to have a dog that has a consistent behavior in ignoring old and finding fresh, or vice versa. Whatever you want to do, it's within the capability of the dog, and as long as it's a rewardable behavior, then why not?
Yeah. And my last question, which sort of has to do with that as well, is when you've got like specificity to particular odors. So where you've got two different species of daisy or two different species of snake, or that are similar but not the same. So our dogs can generalize really well. And then sometimes they're so similar, and we're only looking at very low concentrations or low amounts of something slightly different, which I suppose could be diet. But sometimes even the diet will be the same. So chat to me about that.
My experience with looking for something that had a species that was very similar to it would have been the pythons, but they didn't want, if we got a response to one, it was more valuable than the other. So we were able to separate out, but naturally my inclination, the dogs were highly specific. Now I think it comes from the traits that we select these dogs for, you know, more persistence, high reward value, hunt, purpose bred. They tend to, unless you give them the experience, they tend to not generalize. They get highly specific. For example, I never presented any target for pretty much all my eco projects. I didn't say I had a problem, let's proof the dog. I only went back and proofed the dog if the dog showed me operationally, if this is an operationally trained dog, not a laboratory type dog. So the Burmese python dog was only presented Burmese pythons. Now it was given a generalization range from sex to maturity level, juvenile, adults, blue phase, which are shedding, to maybe neonates that had, you know, just hatched, never been fed out of the egg. But you could present them any other type of python and they wouldn't even throw a change of behavior on it in an operational setting. Now, if I put it in a lineup, of course, you're probably going to see something there. But in an operational setting, they walked over every species of snake and did not detect even a snake within the same genus of constrictor.
Yeah, that's exactly the information that I was interested in. So yeah, it's a matter of so in the training, you're doing more like field-based training as well with them, so that they're seeing them. Yeah.
I was taking an approach of don't create a problem until you know you have it, and knowing that these dogs are highly specific, right? The more experience we give them, more reinforcements we give them with just one species, they're gonna, now, like I said, our odor samples we collected were to set that generalization range, right? I didn't want to get out there and go, oh man, they only find sexually mature males, you know, so that we didn't want that issue. But I didn't present other things. You see a lot of people they'll in a lineup like, oh, okay, well, yes, I'll present distractors or proofing odors, which would be components, like if I handled something with gloves or things of that nature. But I'm not going to go say Everglades racers were present or yellow rat snakes and things. They're going to be in the environment. They could be an issue. So I'm going to take them and put them as distractors. Yes, in a lineup, they may learn to avoid them or show no odor recognition, but in a reinforcement rich environment, then you transfer to an operational environment. They say, hey, there's part of the game. And then you see a change. I created a change of behavior. I created that issue with that individual I might have been concerned with. But from my experience, if I am not doing a research project to demonstrate, if I'm going operational, right, I would only stick with that species and I would stick with that species and that generalization range. Now, if you said there's two in the environment, say an African rock python, Python sabay, or the Burmese python, I would go, hey, I'm going to alternate these. I'm going to combine them together. Then we're going to get a bigger generalization range and that encompasses those two species.
Yeah.
But I see so much of people training, presenting things as to proof the dog off before they need to proof them off. Does that make sense? They
Yes.
create the problem they didn't have if they hadn't tried.
So they've included it as in a sterile environment. They've included those odors and said this is something that could be interesting. And even though
Yeah.
you've not rewarded it in a sterile environment, when they're out there, if their odor's not out there, then they're thinking, hey, but this is familiar, because it becomes a familiar thing, doesn't it?
Yeah, it's probably an analogy we use. You work here in the U.S. is baseball. Like first base, second base, third base means nothing. Home plate, you score, right?
Yeah.
You're not going to see a change of behavior for first base, second base, third base in your set lineup in that sterile environment. But when we get on the field, they go, hey, there's part of the game. There's first base, there's second base. These things are key components for me to get rewarded or to score.
Yeah.
Then you see the change. Then here comes the handler cue. Then here comes the response. Then here comes somebody reinforces. Now you have a problem.
Yeah. And you only need one reinforcement often as well. And then it becomes a long-term problem. Yeah. Paul and Mallory, do you want to add anything to that?
I think, well, that's the third leg of the stool that I talk about in imprinting is generalization. And I recognize that you have to train generalization, because otherwise the dogs, as Bart said, will become very specific. And if you don't want specific dogs, you have to include generalization in the training process. Many, many years ago, when the three-leg stool came up from my experience with landmine detection, because of imprinting dogs so carefully on, those targets that you're looking for. And we trained the dogs on Yugoslav TNT, which was what was being found in the Balkans, where the dogs were trained from. The United States Army, the Corps of Engineers, took 4 of those dogs, and I brought them over to the United States. And we got some American TNT to work the dogs on. And the dogs walked it. It was like they didn't even know that that was a target at all. No change in behavior, 0. And yet the dogs in the UK, they'd done 6 months of training on 0.025 of a gram of TNT. I mean, they were good 100% of the time, etc. So I was very confused by this. And what we realized was we hadn't taught generalization. We taught those dogs to be so specific that even TNT, they would only find Yugoslav TNT, no other variant. And we tried other variants. They had no interest. You had to teach them generalization. And that's why I came up with that, the third leg of the stool, because if you don't teach generalization, which is something sometimes you want, then the dogs generally are not going to generalize unless they're very liberal dogs, and they might chance their arm out in the field. And as Bart said, when you're out in the field, and those dogs are finding nothing, they might chance it and see, what can I get away with? Can I, I've encountered this odor before. What will they do if I give a response? But if you really train the dogs to be specific as a rewardable behavior, then there's a good chance they're going to be like that in the field, unless you don't want them to be.
Yep. Mallory, do you have anything to add?
No, just to piggyback off of Paul, I mean, we saw a similar thing. If you're not explicitly training, you're not going to, at least in the laboratory, in a very sterile environment, we saw tenfold, which has been, published in other species, rodents and humans. It's not very, a very big difference unless you're explicitly training.
Yep. Yeah, we have wind farms here that have to be surveyed for bat and bird mortality, and so the dogs are working to pick up any bat and bird carcass, so any species of bat or bird within the area. So training them is an interesting thing, because you've got a huge range of different species that you're training on, but the dogs actually generalize really well to that. And it's interesting that you could train on a few bird species and a few bat species, and then they'll generalize to everything. It's interesting, Bart, with yours with the snakes, that when you're being specific, you actually still have to give them a huge range from juvenile right through to older as well. And I know go.
I don't know that we did within that species, but it didn't hurt. It hedged our bets that we wouldn't have a problem if there was a difference seasonally from breeding to nesting to when they're separate and thermoregulate, and that's all they have to do.
Yeah. So even like I think even sometimes down to like you were saying with stressed versus non-stressed animals. I think that can make a real difference, and it makes a difference to how much odor's coming off them as well. So I don't know whether there's many studies done on that, but that would be an interesting thing to look at as well at some stage.
I could say I mean, we have it documented. There's nothing published, but I have the training logs and distances and things of that nature, because in training fields, we were doing radio telemetry, and those radio telemetry snakes, they were agitated, right? When they were dropped off, they were mad, and I could hit them from 300 meters. Like the dog show a change of behavior, bracket. I'm talking across ag fields, give a fringe response. I never saw in the first probably 5 or 10 snakes that we were dead downwind of in place. It would odor rec might have been 10 meters, 20 meters max, depending on the orientation of the snake, environmental factors, the weather, humidity, things of that nature. But just from an agitated snake to a operational snake in place, I mean, 300 meters versus 10 to 20 meters was the difference in ability to detect the odor.
Yeah, that's definitely something that I think we're going to be talking about reptiles at some stage in one of our webinars as well, and distances to detection, so like the differences between different targets, and how some targets really give off a lot of odor and some don't. And I think definitely their state, even things like I would suspect that whether they're in prime breeding, if they're giving off their breeding hormones and pheromones as opposed to, in a hibernation state, it's going to make a difference.
Yeah, I'd say a mating ball was one of the hardest. One of the few times that I got a response out of the dog. Most of the time, I didn't wait for a full response. The dog had already gave me enough information. Pulled the dog, pythons over there. But a mating ball was, and the male was actively running from us. We found another one. The dog actually was in a full sprint and slid probably 20 meters in a response. That's how strong the odor was. But yes, all those factors do. And the environmental factors were huge, not just in that project and everything we do. For example, like depending on the target, foggy day in the morning, I didn't get the dogs out until the fog burnt up and the air started moving.
Yeah, environmental conditions as well. Good. Anything to add, guys? I think we've sort of covered it all. I'll have a look at all this, and we'll have a few. I'm sure our viewers will have a few questions as well. So I might at some stage after the webinar, if we've got any questions from our members, I might have to email you guys if they've got any questions that we can't answer. So if that's okay. But yeah, thank you so much. And it's been great. Okay.
Wow, that was.
Well, let me just get there we go. All right. Can everyone hear me? Someone wave. Yeah, good. I'm hoping that we had a few questions, mostly from Naomi and Latoya sent in, that I think we covered it all. I had a few things in mind, but they sort of covered things as we were talking. So I've been looking at the chat as we go a little bit, but is there anything specific that we need to maybe get back to them with? Recorded. Megan Barnes, what if I only care about the presence of an incredibly rare species?
Sonja, I was just responding to some previous comments in the chat about residual training on residual being a slippery slope. Like I think there's possibly some objectives that it might make sense where if you want presence only or you're looking for landing sites or anyway, if you want to talk a bit more about that or send it on to our colleagues. I think there's some various related comments in the chat.
Yeah, one of the things I probably didn't cover all that much was talking about the Gen X vials as well, and things like that. And I think there was a question in here about asking Bart about if you trained on residual and how quick do they go onto the actual animals. And I think certainly that they actually trained quite a bit on snakes, so they didn't train on swabs to start with. Well, I think they did a little bit, and then yeah. Can you talk a bit more about collecting headspace odor? So I think Bart, Mallory, and Paul all have access to labs, so collecting headspace odor, so that they're doing that. But it can also be, I suppose you can, again, you basically just having, if you've got a small thing, having it in a contained space so that it gives off headspace that needs to be enclosed, and then you can get the odor from that. There are a range of different vials and things that we can get. It's Gentex, isn't it, Naomi?
I say get Zent.
It's
Get Zent.
Get
Yeah.
Sent.
Get Zent. Yeah.
Like Get Sent odors. They'll pick up headspace odors, but it's harder if you've got. I mean, you can't put a python in something like that because they only come in little small vials.
We've played around with, like, platypus, for example, having them in their nesting boxes and having the Get Sent tubes wired in the tops of the boxes. So it's not the contact with the animal, but it's more collecting the odor of the animal. But it really depends on the amount of time that you can have that animal contained ethically, and obviously have ethics approval to do so.
Yeah.
So sometimes those can be limiting factors on how successful those collections seem to be.
Yeah, one of the things
we're certainly doing that now with the collet Delma and the little tubes.
Yep.
You can only hold them for 24 hours.
Yeah, 24 hours should be good, I think.
It is.
Yeah.
It is. But again, it comes down to that: are we getting enough scent?
Mm.
I can't smell it, so I don't know.
The beauty of machines, unfortunately, they're really expensive to use.
They are.
But I would think that 24 hours would be really good. We've been limited to like 3 hours for some of our work a while ago, and I was having to rotate individual animals over the course of 24 hours to try and bring it up to that amount of time. That was quite challenging.
Yeah. So hopefully, Rachel, that answered your question about headspace odor. So the Get Sent tubes would be the way to go. You can order them online if anyone's interested. So it's quite a good thing to do. Is there any other questions? There we go. Yeah, so a bit of opinion on the Get Sent tubes. That's from you, Dennis. So you're using those, aren't you, Dennis?
Hmm.
Yep.
There's been some studies on odor absorption with, or maybe it's not been published, but people talking about absorbing headspace odor onto things like gauze swabs, etc. as well, which is something that can be done depending on your target odor in conjunction with other devices like TAD devices. I don't know if anyone's heard of those. But if it's a live animal odor collection, TAD's not suitable.
Yeah. I don't think there's many more questions. Has anyone used Get Sent with turtle nest eggs? If so, how? Sarah Marshall? We've got a couple. Have we got a couple of turtle people here? There should be a few.
We didn't try Get Sent. It was
not
around at the time that we were doing that.
No, I was just thinking Nick's done turtle nest eggs, but it was before Get Sent. And then Rachel said she knows someone who's done it successfully. Yeah, okay, that's all right. So that's good. All right. So recent podcast. Oh, there's a recent podcast on Get Sent. It was quite interesting. Answers some of the above questions. So there's a link there for everybody if you want to have a look at it. Yeah, and then got experience with has anyone got experience with differences between odor from captive animals versus wild? I think there's quite a lot of people that have done that. And Bart was actually talking about that a little bit as well. So anyone that's in our webinar that's had that issue or have got that experience of captive animals versus wild?
We certainly have with koala detection, training on animals that have been in care compared to animals that are in the wild, and even more so just animals that have got different diets up and down the East Coast. So northern New South Wales is completely different to the South Coast, and then Victoria is completely different again. So depending on what the diet is, and it can be as little as on the coast and on the plateau, which is only a couple of 100 kilometres, but yeah, it can be quite a lot of difference.
Yep. I think Trace, did you experience any differences with the quoll work that you did very early on with the captive quolls as opposed to wild quolls?
Well, that's the problem, isn't it? Where are the wild quolls in Victoria?
Yes.
So with that, that was the only option we had. I think when you're talking about those kind of close linkages, I was actually thinking about koala, like what you were talking about before around diet. We had koalas. I was looking, this is years ago, looking for koalas in blue gum plantations, and yet only at the time had koalas on diet with manna gum. So then had to work with a carer to pulse in blue gum into some captive to get that. So I had wild manna, then I had captive blue gum, but presenting those two got the dogs to kind of get that picture. And it's not that the dogs kind of didn't loiter and hang, and you know you had to be on them to let them know, but it can make a difference, because they what color are they picking up in the picture? We're trying to teach them to find red. You know, where is it?
Yeah. We're having trouble hearing you, Trace. I can just hear you. All right. I think we've sort of covered all the questions. Anyone got anything that I haven't picked up from this chat?
Yeah, it's Lynn here. I noticed there was a thing from Megan about whether we'd used the Gettanks tubes with Hastings River Mouse work we've been doing. And we haven't particularly, but we have used a head scent collector that we built up based on a design from Paul, of basically using a vacuum pump over odour and drawing it out onto filter paper. So that's it. I don't know whether you'd call it live odour, because it's collected material, so scats and fur and swabs and stuff. So it's sort of, because we can't get access to live animals to train on. But it proved to be quite a useful technique of just being able to, really basically because it meant we could save the odour that we had in the fridge, and we could actually put it onto filter papers and then use them and put them out in the environment as a draining tool, which I guess is similar to the tube concept.
And have you had success with generalisation from those training odours to live detections, Lynn?
Yeah, we have. The dogs are now operational.
Brilliant.
A couple of weeks ago. But yeah, we're lucky that we're able to move from the captive material from trapped animals to working on live animals on the trap grids, and then to radio tracked dens.
Yeah.
And so we were able to sort of build the profile in the field. And yeah, it's been quite a process.
Yeah, it's all pieces of the puzzle.
But I think certainly if we'd had the opportunity to stick a live HRM in a chamber with the vacuum pump, then making that connection in the field would have been a lot easier.
Yeah.
But poor HRM, so we can't do that at this point.
Yeah, and unfortunately, there's no animals in captivity, so it's you know, I still think if you've got access to a live animal, captive or not, you're ahead of the game than working with individual bits of fur and scats and swabs, etc.
All right, lovely. Thanks to all our members for piping in and answering questions as well, since we didn't have our actual speakers. So that's been super great.
Thanks, Lynn.
Yeah, so thanks, Lynn. Thanks, Naomi. Thank you, Dennis. Thanks, Trace. A couple of little take-homes, things that I found were really interesting, and there was so much in that webinar that I don't know whether you know, I was writing madly the entire time. I'll actually answer your question with this, Daniel, a little bit. He was asking mentioned about the customs experience issues with availability, and would we think that that would be with the TAD? One of my take-homes was the fact that containers and barriers act like a filter and therefore filter out some of the odour. And I thought about that before, but thinking about barriers or the containers that you're holding odour in as a filter, that so therefore the odour available to the dog is only part of the odour that would be normally available as a whole if the container wasn't there. That gave me a really good visual on how containers work. And so I suspect that most of our things like TADs and even GetSense would work as a filter to some respect. So they'll only hold some odours and filter out some odours. So that was a big take home for me, and it got me really thinking. I've had a week ahead of you guys, so I've been thinking, my brain's been going a million miles an hour. Yep. And the other one that I thought that was amazing, because I don't think that anyone's done it yet, is the fact that Bart was talking about dogs picking up odour at 1 part per quadrillion, which if anyone's actually tried to get odours down, thresholds down, it's just the stuff that we do. And I can't even think getting it to 1 part per quadrillion is amazing. And the fact that their dogs were picking it up at that point is also amazing. So that also blew my mind a little bit. So yep, hopefully this has been great for everybody and you've got a bit of take home from this and learnt some. And this webinar's been free, but we've decided that ACDN webinars in the future will be, if you want to continue to watch our webinars, that you will need to be a member. So we will be putting out memberships on our Facebook page and also on our website. So if you enjoyed this webinar and would like to see future webinars, becoming a member is the way that you will be able to do that. So I hope to see you all very, very soon at our next webinar. We'll be having them regularly for the rest of the year, and we've got some really amazing topics and fantastic speakers. So thanks, everyone.
Thank you, Sonja. You did an incredible job and just hit those questions really, really, really well.
Right. Thanks, everyone. All right. We'll head off.
Thank you. Good night.